What I'm going to talk to you about today is a project we've been working on for about 7 years called the Virtual Brain. It's part of an international collaboration amongst about 10 different universities spread across North America, Europe, and as well as in Australia. And there's two sections to the talk. One is primarily focused on theory and application, and the last part's focused more on sort of the next phases for the project. So hopefully it all makes sense to you. If I guess we should wait for questions until after the talk, or during the talk, whatever. So the outline of the talk, basically the first part of dimension is talking about the theory and the architecture underlying the platform. And then some applications. The first application looking at what are called resting state networks in the brain. For those of you who know the neuroscience literature, there's been a lot of buzz right now about what's your brain doing when you're not doing anything. And this is called resting state networks. Has been discovered, quote unquote, using functional MRI. And there's really some questions as to what does it actually represent, these resting state patterns. And that leads into an exploration of the internal noise in the brain, finding that actually the noise in your brain is quite an important feature of enabling your brain to actually explore what it's capable of doing, but also maintaining brain health. And then there's an obvious extension then to the application for understanding brain damage and disease. And then the last part is a new project that's using the virtual brain as sort of a broader sort of social neuroscience experiment called My Virtual Dream. And I'll spend about 5-10 minutes on that, time permitting. So when you ask people how the brain works, you get a variety of responses depending on the profession of the person that answers the question. There's some nice cartoons that represent sort of stylized versions of that. You got the guy plugging in wires in the brain. And depending on how cheeky the person is who's asking how does the brain work, you can say it works very hard. This is generating a lot of sweat. If you go across the history of neuroscience, again, there's a broad range of what people think is happening when the brain's generating cognitions, emotions, consciousness, and so on. And right now, I think the major focus on brain function and how we think it really works is that it's some sort of network property, that the brain is a series of connected systems, and their connections are what mediate different forms of cognition. So now we're talking about brain function as emerging from different network operations, that these areas work together to instantiate different kinds of mental functions. It's kind of obvious in some ways if you look at the architecture of the brain that there's something important about connections. This slide here is actually a real brain. It's from an atlas from Lesion done about 1920. And that's just the blunt dissection of a cadaver's brain. You see the very nice fiber tracks that are coursing around the different parts of the brain. These days, we have the capacity to measure these tracks, at least in principle, in living tissue in a brain in a human when they're sitting inside an MRI scanner, using techniques called diffusion tensor imaging or diffusion spectrum imaging, which is sensitive to water flow through tissues in the brain. And the assumption is that that water flow tells us about the direction of fiber pathways in the brain. So you can actually get something that looks sort of like that from using DTI and DSI data. So you can measure connections, you can measure networks in the brain. And certainly there are now an explosion of methods that are available to help us measure and quantify network operations in the human brain. EEG, fMRI, we can this is an fMRI scan. This is, again, another representation of diffusion tensor scan. And now there's also an explosion of informatics architecture that are used to try and pull these things together. XNAT is one that's been developed out in the East Coast, but there are other variations of these databases that allow one to pool these data with the idea of trying to have some sort of sensical confluence of these things created from these intersections. Analytically, there's a lot of new methods that are evolved, certainly in some of the conferences involved in neuroimaging. About half of the presentations are focused on data analysis. It's been a massive increase in that across the domains. This is a representation of these resting state networks that I'll talk a bit more about later, using independent components analysis. So where are we at with this? I mean, this has been going on now for arguably about 20 years. And the question is, how far have we really gotten in terms of really understanding how the brain works? We can get a lot of data from an individual. It used to be about 3 gigabytes. Now we're getting into hundreds of gigabytes and probably up to the terabyte level in terms of the information you can gather from one person's brain. And that's only in one session. You can imagine if you're tracking a person across time, it explodes tremendously. Arguably, I think the computational architectures are up to speed in terms of being able to pull these kinds of data together, whether you talk about high-performance systems in-house or cloud computing and so on. There are software as well as hardware that are able to at least pull these data in some reasonable way. But the real problem is that we're still focused on single studies, single exemplars of how the brain works. Instead of looking at how does the same thing that's in my head do everything that I can do. I can see, I can smell, I can walk, I can talk, I can eat, I can usually talk, and so on. I sleep, I eat, whatever. All those things happen in my head, in the same brain. But most studies actually focus on a single function. So really the question is, what's unique about the functional and structural architecture of my brain that allows me to do that plethora of things that I can do? And that's where this virtual brain project came into play, is that it provides that means of taking large sets of data and trying to merge them to understand what is it about the function and structure confluence in a brain that allows this plethora of things to operate. This gives you an overview of sort of the workflow for the virtual brain platform. I'll give you a bit more details in the architecture in the next slide. The idea basically is you start with data you measure on an individual. You can get a person's cortical surface. You can get a measure of their connections, for example, using diffusion tensor or diffusion spectrum imaging. Using their actual geometry, you can generate local population dynamics based on derivations that will give you mean field approximations for populations of neurons in each part of the brain. You can parcel it however you want. You can make regions of interest. You can make a large surface where you have a continuously changing populations of neurons. So this represents sort of the underlying architecture in the structural sense. The platform also allows you to stimulate nodes, whether you do it, for example, using TMS, transcranial magnetic stimulation, or stimulation as you might actually see in an experiment. So do visual stimulation, auditory stimulation, and so on. And then generate what we actually measure from your brain, whether it be local field potentials if we can stick an electrode in your head, or EEG, MEG, or generating fMRI bold signals as well. And then it completes the cycle going back then to the empirical data to make sure that the model's reproducing accurately what we've measured in your brain. This is sort of the core architecture of the brain. This is a paper that we submitted to Frontiers in Neuroinformatics. It's under review right now. It should be through the pipeline in about a month or so. The core really is the simulator that takes a wide range of knowledge that's been accumulated over the years about how to measure, how to generate veridical activity from populations of neurons that you actually would measure. So various types of approximations, Wilson-Cowan, Fitz-Nagumo. I can give you a list of the different equations that are in that particular simulator. So there's ways you can measure population dynamics, various forms that you can capture the structural connectivity based on the weights, the distances between nodes in the matrix, how fast the information is transferred, as well as how strong the connections are in the matrix. You have surface dynamics, the generators themselves, the integrators, what's monitored in terms of the data that's generated, EEG, MEG, and bold, and then stimulation parameters. All this then gets written out to either your local computer, or you can save it to another format if you'd like. It's written in Python. Right now the license is the GPL version 2. The platform itself is scalable and modular. I can run it on my laptop. I can run it on a large cluster. I can run it on Unix, Windows, Mac, whatever you want. And it runs under OpenGL primarily. This is a screen capture of the interface. The core of it is the simulator. It's a simulator cockpit that allows you then to set the parameters in this screen, the connections, how the connections are modulated, etc., and then what you want to measure from that model. EEG time series, you have the connection matrix here, some bold data here, as well as some analysis that can be done, principal components analysis, Fourier transform, and so on. That's what you would see if you're generating either cortical surface field potentials, as well as the bold will be generated through this module. You can visualize it. You can also dump it out to a file for data analysis. This is showing you the EEG generation. There's a 32-channel cap on this virtual brain now, generating the cortical EEG signals. These are just some very coarse videos that show you some approximations. The virtual monkey brain is a virtual fMRI study, so we took the macaque essentially and threw them into a scanner, and that would be the activity that you would generate in this virtual monkey brain. On the other side is cortical surface EEG. If you actually strip away the skull and put ECOG, which you actually do, for example, pre-surgical mapping, that would be the pattern that you would get from this virtual brain. And they're pretty pictures too, so you have that kind of aesthetic aspect to it. So the nice thing about this platform is because it's using empirical data as the basis to generate the model, you can go back and forth between the model and the simulation to see what is it that's critical in the model to make my data fit what I actually measure in the individual. So for example, here is some data from, I think there were 100 subjects in this particular one, looking at the anatomical matrix. That's sort of the average across these individuals in terms of the connection weights, the distances between nodes in the matrix, and the directionality informed by some other anatomical databases. And then the overall pattern of functional connections or correlations between time series of these different nodes in the empirical data, and then the corresponding simulated data from that model. The correlation coefficient between these two matrices is about 0.47, so it's not too bad. We could certainly improve that. And down here you see the range of fits that one can get. It's probably hard to see the numbers, but basically there's the range of the correlation values that you can get across different parameterizations of the model. This paper is now, I think, available online in Brain Connectivity Journal. And it goes through this whole process of how you generate these models and do the empirical fits back to the data. That's an example from fMRI. This is an example from EEG. If you do cortical source modeling and then fit the modeled sources to the empirical data, these are some examples of the fits you get. Again, they're quite remarkably close. I'm going to forget which red and blue is here. I think the blue is the real data and the red is the simulated data. So the different cortical sources in about a 1 second snip, and you see the variety of different fits. They're all pretty good, actually. They're rather high, remarkably so, given this is a relatively simple model. But again, the idea is go back and forth now and see what are the parameters that I had to change to get this model to fit my data. And that helps you understand what are the critical features in the brain that allow these dynamics to emerge.
Is it just resting dynamics, or are you stimulating it?
Those are resting state. There's a series of studies right now that we're using somatosensory stimulation, as well as mapping it back to what we see with TMS, for instance. So I'm going to go through now some examples, as I mentioned, in terms of the empirical applications to resting state anatomy, to understanding the importance of noise, and finally the impact of lesions. Is that my coffee? Yeah. Thank you. I have to keep the caffeine levels at a constant, otherwise I'll... So as I mentioned before, resting state dynamics is a big area in neuroscience right now. Everybody and their dog's using these resting state networks for trying to understand what's going on in the brain. At least that's the idea. This is a paper that was published a few years back just showing that across a large sample you get fairly reliable estimates of resting state networks in the brain using independent components analysis. But I would argue still at this point we really don't understand what are these reflecting. Is this simply sort of structured noise? Is it something that's really important in terms of the brain dynamics, or is it something that's just sort of an artifact of fMRI, for instance, which would be a bad thing for the investment in fMRI, of course. So there was some suggestion from empirical work that there's some dependency of the patterns of functional connections, that being the correlation amongst these areas, and the underlying structural connections. So that in some respects, resting state was reflecting what you see in terms of brain connections, the actual structural architecture of the brain. And a very simple study was done by Mike Gracious just up the road, I guess it would be. Finally, there's a fairly good correspondence with what you see between using diffusion tensor imaging and the areas that are functionally connected in the resting state. But it's not perfect, and it's not always direct. It may be sort of indirect connections, which you kind of would expect in the brain where you have both direct and indirect influences. But it's not the case that it's a one-to-one mapping between if there's a structural connection, there's going to be a functional connection. And that's certainly what we found when you start doing the models. And this is a paper that Chris Honey from our collaborative network published a few years back, just looking at both empirical measures between the resting state functional connections and the structural connections measured with DTI, and then taking that same anatomical framework and then generating brain dynamics and finding again that the correlation is high but not perfect. Actually far from perfect, about 0.44. So by measuring resting state networks, you can get some idea of the underlying structural architecture, but it's not completely deterministic. There's something else that's missing in our capacity to understand what the resting state networks are actually reflecting. That's what led to the next series of studies. That while there is a structure-function link, part of the thing that's missing in understanding these dynamics is the fact that the brain is a noisy biological system. But that this noise may actually be quite important in enabling reasonable dynamics to emerge and also to enable a broad functional repertoire of the system to emerge. This is a very simple example showing you the kinds of dynamics you get from a single oscillator when you add noise. With no noise, you ping it, it oscillates and then dies. You add small noise, it doesn't die quite as fast. And if you put a relatively larger amount of noise, it shows you dynamics that you might see if you actually stuck an electrode in that oscillator. If you put these oscillators in a broader network and then scale things like the velocity of the connection and the connection strengths, as sort of a sweet spot in that parameter space, you start getting interesting dynamics emerging that are in this what's called a stability diagram, basically looking at where do you get the transition from quiescence to oscillatory phenomena. And that's these parts of this stability diagram here, right in that sweet spot you see through here. It's kind of interesting because what you find then when you start doing these games with the noise is once it gets to that point of the unstable part of the system, it goes into what's called the metastable state. Metastable state is basically that the system has multiple configurations that it can take at any given point in time, multiple attractor landscapes, to use the vernacular of nonlinear dynamical systems. And what you find is that the system seems to now explore these possible configurations when it has both optimal connection strength, velocity, and importantly, noise. Noise here is very simple noise. It's just basically a white noise added to each individual node. But of course, when you put it in a network, it ends up being multiplicative and much more nonlinear. In terms of resting state networks, the interesting thing is that the areas that seem to be the ones that carry the system through from this quiescent state to the unstable oscillatory phenomena are the ones that are associated with the default mode of the network as well, which are highlighted here in these red balls. And that's a very simple network. Kind of a cool insight, but again, relatively simple way of incorporating function and structure to get you new insights on how these dynamics emerge that we measure in the human brain. We can get even more complicated now and try to get a better handle on what is it that's happening with these intersections between connections, velocity, and noise. Something specific about having the brain at this point in this metastable state that allows it to respond in some optimal way. So we can make a much more complicated model, having, again, a more detailed tractography measure where you get now, instead of 70 or some odd nodes, you get hundreds or thousands of nodes. Generate the simulated bold and measure the functional connections in the simulated data as well as the empirical data. So that's what's shown in this triangle here. And do the correlations across different parameter spaces to see where does this correlation become maximal in the possible parameter space that we can search in this relatively simple model. That's what's shown in this slide, and it's a bit complicated, so I'll just go through it relatively slow. This does say something about having the bridge on the edge of instability. That's been used a lot if you talk about nonlinear dynamics in the brain. This idea the brain is on the edge has been put out quite a bit, but this is actually why it's actually on the edge. This is what we're trying to show you now. So in this column here on the left side of the slide. What I'm showing you at the top is the correlation between the empirical and the functional connections as a function of the strength of coupling in the simulated network. You see as we go across the x-axis that the correlations rise to a peak at about 2.5 in terms of the coupling strength and then drop precipitously after that point. If you measure the phase space of this system, you find that the number of attractors will change as you go across these connection strengths as well. So you go from a fairly trivial spontaneous state where you have a single attractor. As you get to this point where you start having a rise again in the correlations, you get a bifurcation where you get multiple attractors in this new part of the phase space. You get to a peak point, then all of a sudden the system collapses again into the single attractor, which kind of makes sense because you figure if you get to a point where everything is very strongly coupled, you'll have a single system essentially. So where is this sweet spot where you get to this point where the correlation is maximal at that point? What we tried to do now is say, okay, now that's telling us something about where do we actually produce the empirical data as best as we possibly can. There's some peak in that thing. But is there something to do with how the system now responds that makes it want to stay in that particular location? And this slide here, this other column, shows you the response now to stimulation of this model when you go across that part of this space. You find that in this area A, that the response to V1 activation, this is the reaction time of the system, is actually slow relative to if you get to this peak right here where the reaction time goes down, suggesting that the system is actually able to optimally respond to incoming stimuli when it's right on the edge of that point. Okay. So something actually quite important about being on the edge that allows the system to both explore potential states, exploring dynamic repertoire, but also when there is a perturbation, the system can quickly switch and adapt the networks to respond optimally to that perturbation. Now, quickly, there have been a series of studies that I've done with my group looking at whether there is, in fact, noise in the brain that's important in terms of understanding how cognition emerges. So the first thing we found was that if noise is important, it should change as you mature, which makes some sense. What we actually found is that as you mature, the noise in your brain from being kids to adults actually goes up instead of going down. So what I'm showing you here is noise estimates based on single trial EEG data in kids from 8 to 15 and then adults. And the red indicates that there's more variability, there's more noise in the single trial data. So you see that this is pre-stimulus and post-stimulus in an EEG study, that the noise across the groups is actually higher as they mature, and there's actually also a drop in the noise when the stimulus is introduced, suggesting a very deterministic response in the system when you're younger. As you get older, you get a more multi-componential, more complex response in the system. The neat thing is that if you correlate the estimates of noise with performance, you find that noisier brains actually perform better as well in this particular sample. I won't go into detail. You can look at this afterwards, obviously. The top slide just shows you a negative correlation between noise and the stability of your response, stability of reaction time as you're pressing a button in a very simple memory task.
Yep. Do you have similar results for pre and post training for a complex task, like someone's had a year of music training or a year of?
I have it for shorter duration tasks. We're actually looking at not music, but actually acquiring information about a visual representation, and that you find that noise will go up as they learn association, and then as they learn it more, it will reduce in certain areas and continue to grow in other parts of the brain.
What do you call performance?
Pardon me?
What do you call performance?
Performance here, this is I should have given you more information. It's a one-back memory task, so they see a series of faces, and all they're asked to do is say, did that face you see now match the one you saw before? And so the two performance measures were reaction time, variability of reaction time, and the accuracy of the performance in the task. There's no correlation of these measures with mean reaction time. There's a negative correlation to the variance of reaction time, so the more noise, the more stable reaction time is. And also there's a positive correlation with accuracy, so the more noise you have, the more accurate you are in doing this task. It's kind of paradoxical, but it actually, if you think about again, that there's this highly nonlinear system that has to do all this kind of stuff and looking at different configurations, the noise actually might be adaptive in this case to allow you to have that capacity to look at different possible configurations that could ensue. That was in kids. We replicated that in infants as well as other studies looking at kids with different technologies. We're now looking at aging. And for those of us who are aging, it's just to indicate that age-related changes in noise don't always go in one direction. Some goes up, some goes down. And it looks like those of us who actually do have changes in noise in certain areas do better, ultimately, than those who don't. This figure is done using fMRI, in which you find the variance of the bold signal as you age goes down in these medial areas, which are typically, again, default mode regions, whereas the areas in red will tend to go up in terms of their variance with age. But the degree of changes in these areas actually, again, correlates with behavior in this situation.
What are the next on that? Oh, sorry.
Pardon me, sorry. Which
on the right?
This one here?
Yeah.
Sorry, these are age over here, and across here is the standard deviation of a sort of composite score across the brain. So each point is an individual subject.
I'm just wondering why it seems like there's two distinct populations.
There is, because there's no people between 40 and 50 in our sample. Because they work. They don't come in to get scanned. They have day jobs. We also looked at whether the noise scales with task. And we found that individuals in the larger sample, the higher the noise you have, the more you're able to switch network configurations as you go across different tasks. So those who were in the sample either younger, faster, or more consistent in their behavior were also those that tended to switch the noise characteristics of their brain in doing these tasks. Where those who tended to be older or slower or less consistent had less change in the noise profiles with task. Now, I want you to keep in mind that this is within a healthy population. I'm not saying that always having more noise is good. You can envision a scenario where you have too much noise. And what it actually does, that pushes you out of the capacity to have this nice attractor landscape. Or the converse is having too little noise, in which case you have a flat terrain with not much going on. So it's sort of a sweet spot in a sense. Having an optimal amount of noise allows the system to both respond, but also to explore potential configurations, explore what's called a repertoire. Yeah?
Does this mean that what we call noise is not really noise, but it's something that we don't quite understand and that helps the brain work better?
It's a good question, and that's always come up when I give this presentation, is what is your definition of noise? Is it a bad thing or is it a good thing? There's a paper that was published in Nature Reviews Neuroscience a few years back by Wolpert's group talking about levels of noise in the nervous system. You find that because it's a biological system, not perfect, you do have a lot of slop in this system. Imprecisions in channel openings, imprecision in synaptic release, and so on. Whether that's noise in the sense of noise is a bad thing or noise in the sense of an actual evolutionary adaptation, we have no idea. The fact of the matter is, it is this stochastic process that's in the brain that seems to have some functional relevance. And for lack of a better term, we're going to call it noise for now. Okay, applications to patient studies. This is a really severe lesion. This is actually in the virtual brain interface. What we've done is taken a connection matrix and blasted out the entire left prefrontal cortex. So you can see in the connection matrix over here, there's all these gray squares. And then in the 3D representation, there's a chunk of tissue missing. Seldom are lesions that clean in reality, but this is just sort of give you an example of what you can do in the system. Obviously, if you can read in data, you could put in a reasonable lesion, which I'll show you in a second. This is showing you the same representation of the anatomical matrix for the entire brain. This would be the normal case and then the person with the lesion. And if you measure cross correlations this is actually now cortical local field potentials. Again, you see that there's a drop in the correlations for the areas that are knocked out, but you see that the impact of that lesion is quite broad. That there's quite a broad drop in the correlations amongst nodes that are intact as well, suggesting that lesions aren't just going to affect the nodes that are removed, but also broader parts of the network. So to understand what happens in lesion case, you have to look at not only what's gone, but what's left and how that system is responding what it has left to respond to the lesion. And that's sort of the point of some of the other studies we did with this platform, looking at is everything equal in terms of the impact of the lesion on the system? And the answer to the question is no, because there are different capacities for information processing for different parts of the brain because of how they're hooked up, because of their wiring. If you apply graph theory metrics to anatomical matrices, you can characterize different potential properties of different parts of the brain. One of the most popular ways of characterizing it now are looking for hubs in the brain. Hubs are exactly what it sounds like. They're hubs like, for example, the airport at Atlanta is a hub for airline traffic, for instance. In the brain, you have at least two different kinds of hubs. You have hubs that are called connector hubs. And these are hubs that link up disparate parts of the brain. You have provincial hubs, which are connected with areas that have a similar functional operation. For example, visual areas.
We call them state hubs.
State hubs? Sorry. Okay. Keep on forgetting to change my language when I go there. And you have the sort of areas that are sort of primary reasons, for example. What we show on this slide is what the impact is on lesioning these different hubs on the capacity of the remainder of the network to process information. And that's quantified based on the drop in what's called transfer entropy, which is a measure of directed information transfer in the system. You hit connector hubs, you have a massive drop in information processing capacity. This would be sort of an acute lesion. Provincial hubs, you have a much more focal statewide impact. Whereas in the primary areas, the impact is fairly small, if not quite local. This tells us why different lesions have different impacts. It gives us an idea of how to explain that. You can take that and scale it up and actually use a real brain and how that lesion may actually manifest in real tissue. This is someone who had a left parietal cortex lesion. You see here and here the extent of the lesion from a stroke. If you take that and model it in this brain, you see what happens in terms of the change in the capacity of the remainder of the nodes to process information. And what you see happening here is it's not just reductions or increases in information, but actually both happens in the acute phase of lesion recovery. So the blue and red are meant to indicate connections that go up or down in terms of their capacity to process information when you knock out this big chunk of tissue. And again, that's going to help us potentially interpret what happens in the acute phase in terms of stroke, but potentially also gives us a target for modification to help somebody recover, because now we have their brain in the acute phase, and you can envision using something like TMS, androtherapy, and or pharmacotherapy, try and reestablish some sort of equilibrium in terms of the remainder of the connections that are trying to recover from that damage. So where are we going to go next? Obviously, the clinical application is a big area. You can envision quite a broad potential for application for clinical decision support, for instance, in stroke, for pre-surgical mapping in epilepsy. You could take EEG data from an epileptic patient and see, can we predict where the seizure focus might be, for instance, and also predict what's going to happen when you eliminate that node in terms of the capacity of the system to stop generating seizures. As a matter of fact, in the latest version of the software, there's a module called the Epilepter, which allows you to put in stroke or seizure foci into the model. The last thing we're working right now is actually looking at brain stimulation effects, like TMS, for instance, or deep brain stimulation as well. The interesting thing about DBS and TMS, DBS is now used therapeutically, and nobody actually knows why it works. They just know that it does. It's rather typical in neurology, because they're either going to chop it out or stimulate it, and that's typically the way it gets treated. And often is the case, they don't actually know why it works, they just know that it does. So hopefully we'll get a better idea of why stimulation actually works in some cases and why it doesn't. All these techniques, or all these applications right now, are assuming that we're going to take an individual's brain and have that individual brain become part of the virtual brain. But would it not be possible as well to have multiple people actually interacting with the virtual brain now? And that's actually this next little phase of the project. So the virtual brain can interact with one person through for example, EEG, the question is, can we actually do it in groups of individuals? Can we do like a social neuroscience experiment, for instance? And that's this idea of this virtual dream. And the way we're going to do this is it basically based on a large sort of immersive art installation. This rendering here is a dome that, for those of you who know Toronto, this is actually going to be on the edge of the University of Toronto campus in the fall of this year. The idea is that we'll have groups of individuals, 20 people at a time, coming in. We'll have wireless EEG headsets on that will feed into the virtual brain platform, which will be in the center here. And based on the consensus of, for example, spectral fluctuations in their brain waves, that information will then go to modify the audiovisual environment that the people will see inside this dome. Basically, what you're actually doing is essentially creating a dream internally in real time. So the audiovisual fluctuations are going to be a combination of animations whose speed, whose hue, whose vibrancy will change as the consensus power spectra change, and people can actually see this happening in real time. In addition, just to make it even more of a head trip, is we're going to have live musicians using this audiovisual display to cue off of, to jam essentially with the people and with the virtual brain at the same time. So the nice thing is that we can collect these data from these individuals, look at how they're interacting, look at how their brains are trying to drive this thing, see if we can get some consensus in terms of the power spectra, but they also will have fun in principle as they're doing this. It'll be debuted, as I said, in Toronto in the fall of this year. The nice thing, though, is that this platform we're putting together is actually portable as well. So although the 60-foot dome that's going to be may not be portable, the contents themselves can be moved around quite easily in various locations. We're actually surprised to find that there are actually dome vendors that will set up domes for you for a small price. So where are we going with this? So as I said, it looks like we can interact with individual people, but now we have the potential of also having broader sort of social neuroscience experiments. The virtual dream is in fact a merger of art. It's an immersive art experience, but it's also the science underlying social interactions, underlying can we use our brains to actually communicate with one another. It's scalable, it's portable, and we're planning right now to have it installed in various locations around North America and Europe. And it's an incredible amount of data we're going to be able to collect in a single session. Nuit Blanche is 12 hours long. We're measuring probably about 480 people in an evening that will come through this display. In various degrees of intoxication probably, but that's part of doing these naturalistic experiments, of course. And besides that, it's actually kind of fun. And so with that, I'm happy to take any questions. Thank you for your attention.
And a quick one. Would it be correct to think of, we were talking about the question of noise was brought up earlier, would it be correct to think of that in terms of entropy? I mean, it seems like you're finding a sweet spot of entropy where you have the phase states available to the system. If you increase those, then your brain can react better to things.
Yeah. Now, in the network level, actually, I do think we're talking about entropy for sure. As a matter of fact, one of the ways of characterizing this noise at the network level is using a measure that is called multi-scale entropy, which looks at entropy as a function of different time scales. And you find that in fact increases with maturation, positive correlation with behavior, and it changes with age as well. So it's information. If you think about entropy, information is synonymous for sure. But it's not simply, it's nonlinear. So that's the other characteristic of it. But people tend to disregard it as noise, where in fact, one person's noise, one person's information, essentially. That's what we're finding here.
Because the question of, what is noise? Noise generally has this negative connotation, whereas entropy can be,
spun differently.
Yes.
Now, the important point about this as well is that there are different levels at which this will manifest. You're going to have a network level of noise or entropy, but also local levels of noise and entropy. And whether those are synonymous, that's less clear. So it might be that the noise that's at local populations or single neurons may in fact be quote-unquote pure noise. Whereas if you put that noisy oscillator in a larger nonlinear system, that actually may be information. That part we're not actually sure about yet. Back in the back.
back to understanding noise. So does this more noise? Does it might it mean more neurons firing, just more components or larger components? Each of them are have more neurons now that have become adapted to the task or something. While before they were uncaring, they were just firing at the mean rate closest. Could that mean?
No, because if you it could be, but if you can actually test that by, for example, looking at getting rid of amplitude fluctuations, for example, or getting rid of areal changes, and seeing if you do that, does these things still happen? So, for instance, in the EEG data, the empirical data, as well as the subsequent MEG data, what we did is did the same analysis by eliminating differences in amplitude, differences in spectral power, for example, and the entropy differences still exist. So it's not simply a case of having more tissue engaged, it's actually something to do with the interactions amongst the tissue that's really the important part. Yep.
Do you have an idea of the range of signal-to-noise ratios if you're trying to measure something like your EEG system, or maybe better, fMRI? If you're thinking of a note or an image, what's the noise ratio to the signal approximately?
My God, it really depends on, there's so many factors.
But the kind of range of it, how big is the noise is the factor, is it?
I'm trying to remember what it was. I think in the EEG data, it's usually down around 0.2 to 0.3. In fMRI, it can be really crappy. It can be much higher. fMRI, you probably need to do a bit more. It's taking from empirical data down to actually being able to analyze it. There's a lot of things you want to get rid of that really obscure the important noise in the fMRI data. As a matter of fact, and sometimes in the signal-to-noise ratios in fMRI data can be really, really bad in raw data. With pre-processing, you get them down to about closer to EEG, 0.2 maybe.
And that's just the signal, nothing to do with the imaging technology.
Right. I mean, it also depends on how do you define what the signal is as well in this particular case, which is another problem, of course.
Right.
So.
So one comment about noise. It's not really a question. There's I don't know if you know Jeff Hinton.
Quite well.
Machine learning person. He's now with Google, by the way. Very happy now.
Oh.
He's still in Toronto as well. So he did some nice stuff with. I mean, there's been a long idea that noise helps us learn. But he did some recent work, some people in his group, showing that noise done the right way when learning actually gives rise to some very powerful effect that you might call like a mixture of experts. So if what you have is a network, a neural network that's trying to learn, and you zero out certain neurons while you're learning, different neurons at different times, what you get is this effect that in one large network, you end up building an ensemble of less well-connected networks that then vote. It's like a voting mixture of experts that's voting to solve your problem. So it's a nice regularization strategy, and it allows the individual experts to be riskier, so to speak, so to sit closer to the edge. So I don't know how strong of a connection there is, because it's about learning. Yeah. But I mean, it may just be a hand-wavy connection, but I think there's probably something to be said in making a connection between what you're doing and all of the people trying to use noise as different kinds of regularizing and other sorts of techniques.
For sure, for sure.